Well, good evening, ladies and gentlemen. I'd like to introduce Dr. Gareth Banks, who very kindly has agreed to come and give our final lecture in the series this year. And it's on, as you can see, clocks winding down, how circadian rhythms and sleep change as we age. So I think it's a thing which is of personal interest to most of us, on the right side of the curve. Gareth did his PhD in the University of Sussex, studying and categorizing cholinergic proteins in the glial cells of snails. As he said, at least they were compliant. They didn't move about too fast. You could do it. And then went on to study, to work as a postdoc fellow, first of all at the Institute of Neurology in Queen Square in London, then at the University of Oxford, then with the Medical Research Council in Harwell, studying circadian rhythms largely and the biogenetics or the neurogenetics of this. And then latterly, when Harwell closed, he now works with the Institute of
the UK
Dementia Institute in Oxford. So, without further ado,
I forget where I work.
Yes, I forget mine. So, Gareth, over to you. Thank you.
Excuse me. Well, thank you very much. Thank you to the Society for the invitation. Thank you to Malcolm and Richard also for helping organise and set up. Technology is always fun. So, yeah, one of the central things I'm going to be talking about are rhythms, and the risk of making it sound like I want everyone to dance. Life is made up of rhythms. So yes, as I say, we've got life is made up of rhythms. And because we're biologists, because we're scientists, we categorize them mainly by length. We have infradian rhythms, which are long-term rhythms like migration and hibernation that can take place over weeks, months, or years. We've got ultradian rhythms. Things like when you sleep, you go through this cycle of non-REM and REM sleep, which you go through multiple cycles of that throughout the night. But the majority of research are on the circadian rhythms, circa about years a day, so rhythms of approximately 24 hours. And by coincidence, our day-night cycle is also 24 hours long. So just to briefly run through what I'm going to talk about and talk through. I'm going to introduce the sort of fundamentals of circadian biology, how we got to where we got, some key experiments and key findings, and then lean into a little bit, as says in the title, what happens, how they change as we age, as we get older. Because fundamentally, it's something we're all going to do. It's something we're doing right now. A lot of focus is on disease and disease states, which is obviously very important, but not everybody will have diseases, will have these disease states, but everyone will get older, and we're living in an aging, increasingly aging society. And so the understanding of these factors is very important as we go forward. And then I will end with a little bit about sleep. You can't talk about circadian rhythms without talking about sleep. And so although sleep, it kind of deserves its own lecture, I will dip my toe into it. And there's a few recent experiments which are worth investigating and exploring. So start off with circadian rhythms and what they are. So although circadian biology as a subject really has sort of come into the forefront from the 60s onwards, some basic circadian knowledge has been known for the 1700s and 1800s. And studies of mimosa leaves and various other plants have shown, and hopefully the videos work, we did test them, have shown that leaves of a lot of these plants show a circadian rhythm. Apologies, we just have to, there we are. Where in the dark, they collapse down because they don't need to be exposed to sunlight. Light comes on, they lift back up. And they go through these cycles, dark, down, light, up. But what was found out in these early studies, if you take these plants and pop them in a dark room, so there's no exposure to light, this is maintained, and this will happen now. So the next cycle is in the dark. There's no light to switch on. And you can see they still show this. It's slightly for want of a better word, because it's a plant, it's nervous. It doesn't do the full thing, but it is showing this rhythm, and it will happen one more time before the video ends. And these rhythms will persist, and they'll keep going. And that is the core basis of circadian biology. And so those kind of experiments were carrying on for a very long time, but it wasn't until the 60s, this meeting in particular, Symposium on Quantitative Biology in 1960, which brought together people for me, are the founding fathers of circadian science, whose names mean nothing to most normal people. But it was at this meeting that the concept of circadian rhythms was first established, an endogenous self-sustaining rhythm with a period of approximately 24 hours, which can be set by an external cue. So going back to the plant, you have a rhythm of the leaves opening and closing, and this appears to be in response to sunlight, but if you seal it away, the rhythm continues. It's endogenous self-sustaining from within the plant. However, take that plant and move it into a different light cycle, fly it to America, it will start, and then expose it to the light dark environment. Its timing will start to be set by that changed light dark environment. Even though you've shifted its cycle, it will change. So although these cycles are self-sustaining and endogenous, they can be set by external cues. So that's the fundamental basis of what circadian rhythms are. So what does that mean for us? What does that mean to the people? It means that everything we do, our physiology, our behavior, all of this shows a degree of rhythmicity. Although the numbers on this, I'm sure there's going to be someone in there saying, oh no, my highest alertness is in the afternoon, it's not 10 o'clock. This is general, but all of these things show rhythms. Cognition, mood, alertness, pretty much every aspect of metabolism, blood pressure, hormones, even down to gene expression. All of these show peaks and troughs throughout the day. And the obvious question for this is, well, why? What's the point of this? And it comes back down to this 24-hour cycle, this light-dark cycle that we see in the day. The night is dark and it is cold. Day is warm and it is light. So way back when we were little scuttly things on the plains of Africa, back in evolutionary time, we could forage and we could avoid predators in the light easily. We could see, and it was warm. It was good to sustain us as organisms. However, in the dark, we can't see to forage. We can't see the predators that are creeping up on us. And so the best option is to find somewhere and hide and stay warm and try and stay warm because it's colder out there. And that's the basis of rhythmicity, that you have an activity pattern where you're active and foraging in the light and hiding in the dark. And this then follows on, because once you've got this established, you have things like where you need to synchronize your internal rhythms to this. So in the dark, you don't need a high metabolism because you're not going to be getting up and running around too much. So it benefits the organism to lower their metabolism and keep things in stasis, because then you can use all that energy that you saved in the dark in the light. So the circadian rhythms exist because we need to predict these changes. And then once you've predicted those changes, it affects all of your bodily systems. And so this is going right the way back to evolutionary history, right the way back to the beginnings of us as organisms. And so this is why it's continued as we've evolved, as we've grown into humans. So then, given the vast number of things which circadian rhythms seem to influence, what if you muck them up? What if you disrupt your circadian cycle? Well, a lot of things go wrong. Sleep loss, it affects your thinking, your cognition, your mood. Your metabolism is affected, so you'll have weight gain or potentially entering things like pre-diabetic states. You'll see suppressed immune function. You'll see associations with diseases such as cancer or neurodegenerative conditions. So this part, I'm summarizing decades of work and hundreds of papers. So I'm not looking at everyone and showing you all the data for this. But what it is worth considering is, how did we get to this? How did we establish these facts? And so we did this in two ways. First of all, by our animal models. animals are straightforward. You can take an animal, and circadian biologists like rats, mice, and hamsters, because they are small and they are easy to manipulate. You can put them in different light cycles, or you can give them access to food at restricted times. So you can give them food in the day when they should be asleep, forcing them to get up to eat when they should be asleep. And when you track their movement, so the black bars represent activity on a running wheel. This is in a mouse. You can see certain environments they can adapt to. Here, they maintain rhythmicity in these changing environments, whereas here, they're unable to adapt to this light cycle. So you can take these animals and say, okay, how does an animal which can adapt compare to an animal which doesn't adapt? Once you put them in these conditions, then you can take them, you can put them into your behavior, you can put them into your blood pressure monitoring, you put them into metabolism monitoring, whatever you want to do. Can we do the same in humans? Sort of. There are historical, and to be fair, they're still doing it, some studies where humans are locked in bunkers without access to light and then allowed to get on with things as they see fit. These, although these are useful, you can only do 1 or 2 people at a time because you've got to monitor them just in case they get sick, and they're actually quite expensive. But we can get groups of people because as a society, we create some of these circadian disruptions. Jet lag, you fly to America, that's 9 hours difference. Your body still thinks it's 9 hours forward. I always forget which way around. Your brain knows, okay, no, it's light. I need to be eating food now, even though your body thinks you're asleep. So jet lag's a fairly common one, which a lot of people establish. A more recent concept is social jet lag, which is where your socialization and your social contracts, I guess, force you to do things again, which is against your clock. This is often representative as young people partying until 2 or 3 in the morning. I don't remember. Anyway, but more often we're increasingly seeing this as work as well. Particularly now we've got things like working at home and the ability to adapt our work regimes to better suit our work-life balance. So a lot of people are sort of catching up at work, where late at night you'll be slumped in front of the computer because you've had to deal with kids in the morning or something like that. And so these are all examples of these social jet lags where you're pushing your active. You keep your brain active when actually your brain should be saying, no, I'm falling asleep. But then the main one we have is shift working. People which have to work at night, security guards, nurses. I feel genuinely sorry for nurses for many reasons, but 80% of the studies I see on shift working are on nurses because they're very useful because they're already at the hospitals that you can do your studies on. So not only are we subjecting them to these chronic jet lag conditions, chronic shift working conditions, they're then the guinea pigs to tell us how badly those conditions are affecting them, like they don't suffer enough. Anyway, so this is how we've reached these conclusions about how these shifts in circadian cycles are affecting us and how that big list got formed. But how is this happening? Well, the short answer to that is it depends on the system you're looking at, but as a sort of principle of concept, as a sort of a way of showing it, I picked insulin and mistimed meals. So during the day, insulin is readily secreted and you are very sensitive to insulin. And this is good because you are primed to be eating during the day. And therefore, the response to the meal is insulin will be readily secreted, you're sensitive to it, and that will maintain your blood level, your blood sugar level at a reasonable level. And you want that because if you have elevated blood sugar levels for an extended period of time, you start to lose insulin sensitivity. And this can all lead into pre-diabetic states and things like type 2 diabetes. But this is what happens when you have a meal at night. At night, insulin secretion is suppressed and sensitivity is lower due to things like reduced numbers of receptors and things like that. Therefore, if you have a meal at night, you end up with a prolonged elevation of this blood sugar level. And as I just said, this will lead to insulin insensitivity. And too much of that will lead into, as I say, type 2 diabetes, pre-diabetic states, metabolic disorders. This is all easy to say, okay, eat at this, eat during the day. But if you're in a condition where you are forced to eat here, say you're on a shift work, you don't have a choice, you have to eat here, it's the only time you've got to eat, or your body clock is set here, even though you know, socially you are here, so social jet lag or normal jet lag. This is where this mistiming can come in, and this is where you have damaging deleterious effects which are going to come in because of this change in sort of circadian rhythms. So that's your broad brush introduction to circadian rhythms, what can go wrong, how they affect things. What happens when we age? Guess what? It starts going wrong. You probably could have guessed that. That's what happens when you get older. The short story is all the rhythms we can measure, and there are a number of things we can measure with hormone levels, temperature, you know, things like these are relatively straightforward. Everything starts to fragment. In terms of the maths, if you're looking at it as a sine wave, you're looking at it as a curve, you see a reduced amplitude. The difference between the peak and the trough of your rhythm shrinks. This sometimes is because you actually lose some of these absolute values. This is what we see in gene expression. Genes are less able to be expressed, and therefore you see a reduction in the total amount. More commonly, we see fragmentation, and this is most commonly seen in sleep. When you're, I was going to say when you're young, no, children are weird and different for sleep, we'll ignore them. When you're in your 20s, you can sleep very solidly. You'll have a lovely, prolonged period of sleep all through the night, wake up, prolonged wakefulness all through the day, be active. As you get older, you wake more during the night. You start to nap during the day. This is what happens with these rhythms. You start to, the things which you should be doing in the rest phase, you do in the active phase, and vice versa. And this is the fragmentation of rhythms which comes through. The other thing which happens, you might be able to see with some of these graphs, you see a shift. They start to shift to earlier phases. And this is being very widely studied with the concept of chronotype. There's a chap called Till Ronenberg, who's one of the leaders in the field who studied this quite intensely. And this is your morning person or your evening person, your lark and your owl. We all know that kind of person who gets up and bright and early at 5 in the morning can just get on with stuff. And we hate them for it. But this isn't constant with age. So this is how they score it. So the higher up you are here, you're more evening. The lower are you here, you're more morning. You start off and you have this rapid delay. So by the time you hit late teens, early 20s, you are at the most evening type. This is where that stereotypes thing for teenagers comes from. Come on, get out of bed, school's starting. And much as though there's many reasons to hate teenagers, in this situation, it's not actually their fault. They are being, their genetics is pushing them to that. They are pushing them to be later during the day. But then when you get older, this then starts you start to lose this. And by the time you've hit, 50s, 60s, you've shifted dramatically to this morning type. And I've got to admit, I'm feeling it. I could lie until 10. These days, I'm up at 6 or 7 in the morning. And so this, by personal experience, this happens. This is one of the most replicated findings out there, that we have this shift as we get older. As we get older, we have this shift to an earlier and earlier state. And so just going back to this consequences of circadian disruption, when I first entered the field, everybody was very excited about, okay, look, circadian disruption can affect psychiatric, neurodegenerative, metabolic things. Is this affecting all these diseases? But then due to projects I was working on, I started looking at this and saying, wait a minute, sleep loss. Well, that happens. You lose sleep as you get older. When you get older, you don't think quite the way you did. You have cognitive defects. Mood, I think I need to consult a psychologist, but I'm not sure about mood. But you certainly gain weight. There's suppressed immune function, and you're more susceptible to things like cancers, neurodegenerative conditions, and metabolic disorders. And so in my mind, I started linking the two, saying, okay, is there a link between this disruption and sleep? And much as though I thought I was brilliant, it turns out in the 70s, these founders had already thought this. So yeah, I didn't get there first. So this is a paper by Colin Pittendrow, and I can't remember her name, two again of the founders of circadian rhythms as an academic approach, outlining circadian resonance as a concept. Organisms perform most effectively when, as oscillating systems, they are driven by external cycles close to their natural frequency. In less flowery language, if your internal rhythm matches the external rhythm, that is the most healthy condition. That is what they're proposing. And they showed this, their initial findings were based on findings in Drosophila, the fruit fly. And Drosophila lived longer. They had a series of mutants which showed different circadian periods. And the Drosophila lived longest when they're in conditions of 24 hours. When their circadian period was 24 hours, which matched, of course, the 24-hour period light cycles that they would have in the labs. And this has been followed up by other correlative studies, which have looked at a lot of laboratory animals. So these are different mouse strains, these are different primates, and these are different rodents. We have a lot of data on these, including circadian rhythm and including lifespan. And if you match the two, there's a strong correlation that the further the endogenous circadian period is from 24 hours, the shorter the lifespan. Correlation, certainly, but this doesn't necessarily mean it's necessarily true. The problem is it's tricky to test, and by tricky I mean expensive in terms of animals, but it's quite cheap to test in bacteria. So this is an experiment back in the 90s where there was where these researchers mixed different types of bacteria which showed different rhythmicities and then exposed them to different light cycles. So initially you had roughly 50% of each type of bacteria. They were then exposed to a 23-hour light cycle. And this top graph, after 27 days, this top cohort, basically this species of bacteria with a period of 23 hours, matching this 23-hour cycle, completely outcompeted the wild type with a period of 25. In this condition, the wild type had a period of 25 hours compared to this one, this strain with a period of 30 hours. And although it wasn't completely wiped out, it definitely favored the wild type, again, closer to its endogenous rhythm. And similarly, the 23-hour one beat the 30-hour one there. And then if you flip that around to a 30-hour cycle, again, the wild type outcompetes this strain because, well, and the theory being that it more closely matched the endogenous circadian period. So this is, albeit in bacteria, but it's a nice little illustrative example of what they mean by circadian resonance, that if you maintain an organism in cycles which closely match its endogenous cycle, it's much better for that organism. And we have had some studies in mice which look at this as well. So this was a study which came out in 2020 and annoyingly beat us, but we'll get to that in a second, where this, you may recognize this from previous slide. They had mice in 3 different conditions, 2 of which they were capable of entraining to. You can see that they can match this circadian rhythm very well. They can't match this circadian light cycle at all. But then when you look at the survival of these mice, the mice in this condition, this advanced condition, they started dying much quicker than the mice in these conditions. We were also performing a similar study, albeit with less harsh light environments, where we had animals which were in dim light and dim light at night cycles. So rather than lights on, lights off, it was lights on, lights dimmer. And then we also had what we called social jet lag, where it was a weird sort of truncating, expanding light cycle. And once again, the animals could entrain fairly well to this social jet lag environment. You can see a period of activity, period of inactivity, compared to the dim light cycle, where it was just much more fragmented. And then when we did our behavioral experiments, and in this case, a bit of neuroanatomy, we saw changes specifically in this dim light at night condition, specifically in the condition where they could not entrain. So they could not adapt to the cycle. Again, linking in the possibility that if you can't adapt to the cycle, or if you have problems adapting to the cycle, it's deleterious to health and aging as you go through. So we've got these things saying, okay, something is going on, linking the circadian system and aging. So the next question is, well, what's happening to the circadian system as you age? And this is a very crude diagram of the circadian system. You have a core circadian oscillator which ticks over. This is what maintains the drive, maintains the cycle. This then feeds onto clocks in peripheral organs and maintains the synchronicity of the body. And then you have an entraining signal which will set the cycle based on external environments like dark cycle. Very basic. In reality, you've got multiple layers of inputs. Then you've got multiple layers of different target organs, and all of these are feeding back onto each other at random intervals. ignore all that. We'll stick with this for now, but just so you know, it is an oversimplification. So what is this core clock? Well, this goes back to experiments in the 50s, where lesions were given, that's the wrong word. Hamsters were subjected to lesions of different parts of their brains. And they found that following a lesion in the hypothalamus, some of these hamsters would become arrhythmic. So here's a hamster with a nice active, inactive, active, inactive rhythm. Following the lesion, which was here, this activity rhythm just drops off. And so more and more of these lesions, more refined lesions, identified two very small nuclei in the hypothalamus, the suprachiasmatic nucleus, as this core master oscillator. It's a twin nuclei of about 10,000 neurons each. These days we have other technologies. So this is something a PhD student of mine was running, where she was able to genetically ablate. So she was able to genetically stop specifically the SCN from developing. So rather than actually going with surgery, she was able to use a combination of genetics to actually just switch off the neurons which would turn into these SCN neurons. And again, we see the resulting mice are arrhythmic. We have some, this is widely known. The SCN is this core master clock, but it can also restore rhythm. So you can, if you lesion out the SCN, you can restore rhythm with a transplant, but you can go beyond that. So this is experiments done in hamsters, two types of hamsters, wild types, which show a nice, almost 24 hours rhythm, and this mutant hamster, a tau hamster, their activity cycle starts earlier and earlier and earlier every day. And just as before, lesion out this SCN, the hamsters go arrhythmic. But if you can then restore rhythm with transplants, but if you transplant a wild type SCN into a tau hamster, you see that the rhythm that is restored more closely matches the wild type rhythm. Similarly, if you pop a tau SCN into a wild type hamster, it's not complete, but it does more closely match the rhythm of the tau hamster. So the SCN activity rhythms match the graft. They don't match the host. So the SCN itself is enough to maintain that clock, to maintain that rhythm and keep it going. And then the SCN itself controls the rest of the body via hormonal outputs and neuronal outputs. There's a lot of those, we don't need to go into them, but just so you know that the SCN then controls things via lots of outputs to lots of the bits of the brain, lots of the bits of the body. But the question is, well, how does the SCN keep time itself? And this goes back to 2017's Nobel Prize, which went to these three gentlemen, Jeffrey Hall, Michael Rosbach, and Mike Young, who were responsible for the identification of the genes which underpinned the circadian clock. Initially, they did this in the fruit fly, but then this was found in many other organisms. Just to run through this, the story of that is wonderful. I would, if you're interested, just go online. The story is out there after the Nobel Prize. But what they discovered with this core clock, so this is your cell, you've got a nucleus. In the nucleus, you've got your DNA. You've also got these two proteins, clock and BMAL. And these two proteins bind to E-boxes, which are elements on the DNA which regulate transcription of genes. When they bind, they initiate the transcription of many genes, but in particular two genes, period and cryptochrome. So the mRNA is translated into protein in the cytoplasm. These two proteins come together, and this re-enters the nucleus. And then these two inhibit the function of clock and BMAL. So without the function of clock and BMAL, there's no activation, there's no cryptochrome and period being transcribed, there's no cryptochrome and period protein being produced. Therefore, clock and BMAL are reactivated, and the whole thing starts again. And strangely enough, this has a period of 24 hours. This to me is one of the reasons why I love circadian biology. Slight sidetrack. Richard Dawkins writes a lot of things, whether people agree with him or not. But one of the things he has written is about he finds wonder in nature, and to me this is exemplary to this. We look at these clocks which have been delicately crafted with these gears and hands going, and this is going on in cells in the body, and the evolution beat us to it by thousands of years. This is wonderful to me. This is why I got into circadian biology. Anyway, sorry, sidetracking. So the core concept of this transcription translation feedback loop, although the proteins and the genes change, it is common to many other species. So you've got different names, but the idea of proteins which activate their own, which inhibit their own transcription, this whole cycle taking 24 hours. This is how clocks work across kingdoms. And this is again why they got the Nobel Prize. And we can visualize this. So this is a transgenic mouse that the period gene is actually also activating a fluorescent tag. And so you can see this cycle of gene transcription and translation going ahead. And again, although this is speeded up, this is approximately 24 hours. So again, what happens to the SCN when we get old? Once again, it starts to break down. There's a lot of studies on electrical oscillations in the SCN. Again, we can see this reduction in amplitude, the difference between the peak and the trough in the young and the old, it gets smaller. The reason for this is, for want of a better phrase, the actual recordings are messier. Again, you have firing when there shouldn't be firing, and you have lack of firing where there should be firing. Again, it's this fragmentation of the rhythm, similar to what I suggested with sleep. So what's causing, oh, sorry, no, before I mention that, something which obviously people like to do, as I mentioned it, is these SCN transplants. And they've done something similar. So you take an old hamster, I believe, in this situation, and you can see rhythms have been very fragmented. And then you graft on, in this case, a piece of cortex, in this case, a piece of SCN, a young SCN or a young piece of cortex. The cortex does very little, but the SCN restores rhythm pretty well. So again, the SCN is able to endogenously generate these rhythms. Interestingly, the animals that had this graft SCN survived longer. Why? Well, that probably goes back to the circadian resonance thing. Annoyingly, this has not really been followed up on, but this is something which, again, I find fascinating. All the other conditions died, as they should do. They have a normal lifespan, but repairing the rhythms, increased lifespan. So what is going on with the aging SCN? Well, the obvious target is, well, these clock genes that we have all these awards for, are they being shut off? Are they not working anymore? And this is something that some people have looked at, but as technology has moved on from how it was initially looked at, people look at specific genes using a technique called qPCR, which will quantify the amount of things in genes. These days, we look at the whole of the transcriptome, every gene that's being transcribed. And we generate plots like this. This is a volcano plot. Very crudely, anything above this dotted line significantly changes in our two conditions, in this case, old versus young. The red dots are all the clock genes or clock associated genes. And the majority of them are not affected by our aging condition. And this is work I've been doing and been trying, and I'm busy trying. Oh yes, okay, so pinch of salt, it's not written up yet, so not peer reviewed. Take it or leave it if you want. But we then back this up and showing there is no significant changes in a lot of these genes by, with other animals with another technique. And we're not alone in doing this. So this was also looked at in macaques. They did a whole transcriptome study and they saw no significant differences in all these clock genes. So whatever is going on in the aging SCN, to break it down, it's not the clock genes. So all of those are fine. The clocks are still ticking over just fine. So what's going on? Well, the joy with these kind of studies is you can take all these genes are changing, and you can look to see if there's any functional enrichment in certain types of these genes. And these red genes here are all genes involved in synaptic transmission. And just to bring, I've not really mentioned synapses or neurons, but just to sort of give a little bit of a 101, your brain is made of neurons. Neurons are very specialized cells, which stretch out all these projections, and it is via these projections that they communicate. And this is the neural network. This is how the computer of the brain works. You have communication between these cells, electrical signals firing between these cells. Where they form these connections is the synapse. The synapse is a very specialized cellular structure, where two neurons come together. You have a presynaptic neuron which releases neurotransmitters which bind to receptors on the postsynaptic neuron, and this propagates the signal to carry on the electrical signal to other parts of the brain. All those genes I just highlighted in red were found here. Their function is to allow these vesicles to bind and release neurotransmitter into the synaptic cleft. So what appears to be, and they're all downregulated. So what appears to be happening in the older SCN is the neurons are no longer as capable of firing their neurotransmitters. They're no longer capable of communicating particularly well with their neighbors. And that's important because if you're maintaining a clock, it doesn't matter if everyone's got their own individual clock, because eventually they're all going to run out, they're all going to start running at their own speed. You need to have a network so the clocks can form a cohesive whole and actually form a cohesive clock. And you're losing this cohesive clock by losing this communication here. So this is what we believe is happening with the aging SCN, why it's breaking down. Not that the clock is failing, the clock within the cell is going fine. The cells now can't communicate with each other to tell each other what time they think it is. Minor tangent, but something else cropped up with this, and this is kind of more exploratory. There are a large number of genes which are associated with aging. They've been known as biomarkers of aging. And so when I had this data set, I had a look for them, and none of them were changing in our aged SCN. So I looked a bit more closely. So if you look in the cortex, these two genes are thought of as biomarkers of aging. And in the cortex, you see a steady increase in expression as the cortex gets older. Sorry, that should not be there, doesn't matter. In the SCN, it is basically flatline, suggesting maybe the SCN's aging slower than the cortex. So I looked at another method of looking at this is looking at methylation of the DNA. As you get older, the DNA in all of your body starts to accumulate methylation, starts to accumulate these methylated sites along it. And these have also been correlated with aging. There's Steve Hobarth and Ken Raj have generated these chips where you can tell the chronological age of someone just from a blood sample by looking at these methylation markers. Annoyingly, there's not enough DNA in an SCN to do that, but you can pick. So I found one of these sites which had been shown in other animals to actually be representative. And again, we saw there's a reduced methylation in the SCN compared to the cortex. So all of this is just leaning into the idea that actually the SCN might be aging slower than the rest of the brain. This isn't really going anywhere. This is an observation, but it's a curious one. And it's not unprecedented. The cerebellum, so this is now using those epigenetic clocks that I mentioned. Using that technology, they've demonstrated the cerebellum ages slower than the rest of the brain. So it's not like differential brain aging is completely novel. So it would potentially, yes, there might be something in there. Mm-hmm. The other thing is that video I showed with the oscillating expression. The current record for maintaining one of those in a dish is, I believe, 15 months. By changing the things, they've managed to maintain those oscillations. That brain slice kept alive for 15 months. I think the best we were able to do with literally any other organ was the liver, and it lasted about 3 weeks. Something which Mick Hastings at the LMB in Cambridge has often stated is he believes the rest of the brain is killing the SCN because we can't maintain the rest of the brain in the same way we can maintain the SCN. So there is some evidence that maybe the SCN has a differential relationship with aging than the rest of the brain. As I say, this is all based on observation. It's not going anywhere at the moment. It is a curious observation which has come out of some of these studies. So back to the circadian organization. So that's the core pacemaker. That's what happens when that gets older. What about this entraining signal? What's going on there? Well, the main entraining signal we have, the main thing which sets the clock is light. And obviously light is detected by the eyes. You probably don't need me to tell you that. We have rods and cones in the eyes with color and black and white vision. But curiously, there were studies where certain populations of blind people were shown to be able to entrain to a light-dark cycle. So here's your normal subject. You've got melatonin, which is a light-induced hormone. which both in and the normal and the blind patient, I'm not sure I like the word normal, but it's an older study, the two showed the same entrainment and showed the same light responsiveness. And yet this patient's blind. So how is this light information getting there? At the same time, groups led by Russell Foster in Oxford were showing something similar in mice. So these mice lack rods and cones, and yet they still show circadian responses. So something else is going on, and eventually a lot of molecular cloning. The answer was found to be a gene, a novel photoreceptor called Melanopsin. But rather than being found up here with the rods and cones, this Melanopsin was found here in these retinal ganglion cells. Again, this is the retina. You have signals from the rods and cones which are propagated by the bipolar cells into these retinal ganglion cells, which then go by the optic nerve up to the brain. The Melanopsin is expressed here in these retinal ganglion cells, and these are, and rather than giving light information to visual processing centers, these project directly to the SCN amongst other regions. And so this explains what happens in these patients. So in an intact retina, light hits your rods and cones and hits Melanopsin, and that sends signals up to the brain, fine. But if you lose rods and cones, you've still got these Melanopsin cells working. And so light signals can still go directly to the SCN. Melanopsin does not go to the same places, doesn't go to the visual cortex. So you're still blind, but you'll still take in the information which will allow you to entrain to a light-dark cycle. It's only if you lose everything, which you will then have no signal reaching at all. So it depends on the reason so why people are blind. And Melanopsin, these different rods and cones all have different spectral sensitivities, will be different, will all take in different wavelengths of light. Melanopsin is at 480, and it's from here we have these blue blocking effects which have come on. You may have seen them on your phones or on your computers. There are options in the evening to switch off the blue light. It's a bit of a false flag. You have these things, oh, okay, I've switched off the blue light on my phone. Right, it's fine. You've got the TV in front of you, you've got your reading app, you've got your light inside. It doesn't work. What does have some evidence of working, these blue blocker glasses. So these you can wear, it blocks out all the blue light. And there is a number of studies which shows improvements in sleep and things like here where you show improvements in expression of melatonin, for example. So these, although I don't believe they've gone through full clinical trials or anything like that, but there is a lot of evidence they do have some effect. There is anecdotal evidence that there is a decreasing effect with age. Again, I don't know of any specific studies showing this, but I know there is anecdotal evidence out there. Sadly, the reason is very simple, that these retinal ganglion cells are lost with age. So this is the rat, but we're seeing the same thing in humans. So as we age, we lose these retinal ganglion cells, so we'll lose this input. Again, it's another loss of input to the SCN. So the SCN, as you age, you've already lost this network connectivity. So your rhythms are fragmenting already, but then you're also losing the input from your light cycle, something which would entrain you, something which you could in theory use to compensate for this loss of rhythm. You're actually losing the sensitivity you have to that as well. So sadly, that's just another aspect where everything is breaking down and it's not working very well. Which isn't necessarily good news. Right, time is creeping on. So as I said before, you can't really talk about circadian rhythms without talking about sleep. And I'm going to dip my toe into this a little bit and then talk about a specific study or series of studies, just because it's making news a little bit recently. So sleep, as a lot of sleep scientists who I would count as friends are very, very keen to point out, is not just a circadian process. They get very irritable if it's just all lumped together. Sleep has a strong circadian component. So this is the model of sleep which was proposed, I believe, by Borbley back in the 80s, where you have a process, the circadian process regulates the timing of sleep, but you also have a sleep homeostat known as process S. This is a very posh, a tricky way of saying the more you're awake, the more your pressure to sleep builds up, which is what we all experience. And then the optimum time to sleep is when you've got this build up of this sleep homeostat, this build up of the need to sleep, combined with a low pressure to stay awake from the circadian system, and then you fall asleep. And then the sleep homeostat falls, the pressure to wake the circadian system increases, and so you have the sleep-wake cycle. Unlike the circadian system, sleep does not have an SCN. Sleep does not have a single core section of the brain which regulates it. You've got a lot of different regions, the Velpo, all these nucleuses which have very long names, nucleus ceruleus. All of these perform different functions, but broadly they can be categorized as those which establish wakefulness and arousal, and those which establish sleep. And the balance between the signals is what will maintain whether you're awake or asleep. If you've got more signal coming out of the arousal producing nuclei, you'll be awake, and then that seesaw will drip back down to the other side, and then you'll be asleep. So sleep as a model could be more thought of as this. You've got the circadian pacemaker, the homeostat. There is an influence of social timing. There is the light-dark cycle, because melanopsin cells will also project to some of these regions in the brain. As we get old, it breaks down. That should be the byword for this whole talk. Not only we know the circadian system breaks down, but the sleep homeostat will also start to break down. So the pressure for sleep will start to drop as you get older. And this can also be found in some of these molecular correlates for the sleep homeostat, say adenosine. So the levels of adenosine and the adenosine receptors will fall. And also the communication between these different nuclei which regulate sleep, that starts to break down. You lose synapses and you lose neurons which are connecting all these regions, and that means it's less balanced between your arousal and your sleep promoting areas. So that's dipping the toe into sleep. What's made a few headlines recently dips now into age-related diseases, specifically Alzheimer's disease. Previously, I've just looked at healthy aging, but obviously aging is associated with a number of diseases. Alzheimer's is one of the more common ones. It's the most common form of dementia. And sadly, again, because of the aging society that we are living in, we'll be aware of many, many more people who will have Alzheimer's or be showing Alzheimer's symptoms. At a molecular and cellular level, it's associated with these plaques of the protein amyloid beta, and also these fibrillary tangles made up of a protein called tau. Exactly how this fits in with the pathology of Alzheimer's is still up in the air. While we know these are toxic to neurons, they are often found subsequent to some symptoms, and certainly in our animal models we will see symptoms developing first and then these plaques and tangles, but they are certainly part of the pathology of Alzheimer's, and they are very important to Alzheimer's. So their study is something which, well, is happening a lot. And about 10 years ago, a study came out that looked at amyloid beta levels in the cerebral spinal fluid. So that's the fluid which surrounds the brain and protects it and also helps maintain it. And in both humans and mice, important for mice, I'll say in a second why, you see this oscillation. In the light, you see a reduction in mice, you see a reduction in the light, an increase in the dark. Humans, inverted. In the dark, you see an increase. And this, at first glance, because I probably primed you for it, looks circadian. But actually, again, the joy of having it happening in mice as well is you can actually test things with them. And they subjected the mice to sleep deprivation, where they kept them awake for an extended period of time. And when that happened, this clearance, this drop, ceased. It maintained these levels, and you saw a significant difference if you sleep deprive the animals, suggesting this is not a circadian effect, this is a sleep effect. If it was a circadian effect, you wouldn't see this stabilization here when you sleep deprive them. Further analyses demonstrated that there was a strong correlation with this loss and specifically non-REM sleep. So again, suggesting this is a sleep effect, not a circadian effect. And so this was an interesting observation, but exactly how it came through, but exactly how it was linked in, came into play when we considered this, the glymphatic system of the brain. This, I hope this works. So the glymphatic system of the brain is a way of clearing out the brain. So what happens is the cerebral spinal fluid, there's a flow of cerebral spinal fluid which goes across the brain and clears away metabolic waste products, cellular debris, things like that. It is controlled not by the neurons and cells, but by other cells in the brain, supporting cells called astrocytes. Specifically, they're controlled through aquaporin 4 channels, which are these little protein channels which allow water to flow back and forth. And you can visualize this in humans. So this is just in, this is an MRI scan. And initially we see oxygenated blood, which leaves the brain. And then here, this is the ventricle. This is where a lot of the fluid goes to. It's part of the flow system, I guess, and it flows into here. So this can be visualized in the brain relatively straightforwardly. But, oops, we don't need that again. It can also be studied again in mice. And studies in mice where they were able to put tracers directly into the brain and then trace via multiphoton microscopy. They would trace the flow, and they found that the glymphatic flow is stronger during sleep. And then when they then loaded up these mice with amyloid beta, so they injected radio-labeled amyloid beta into the brain, the clearance of this amyloid beta was quicker during sleep, the same time where this increased flow was occurring, suggesting one of the functions of sleep, one of the hitherto not really studied functions of sleep, is actually this metabolic clearance. And this clearance will also be getting rid of some of these toxic proteins. Exactly how and why this is occurring is still being studied. But one thing is potentially pertinent. There was a study, I'm not entirely sure, but probably about 10 years ago as well, which looked at the ultrastructure of the brain. And this goes back to those synapses, where you have a presynaptic and postsynaptic presynaptic and postsynaptic site which comes together. But during sleep, they drift slightly apart. During wakefulness, they come back together. And so again, during sleep, this drifting apart may create the space which allows the flow to then sweep through and flush out these proteins. This may be particularly true of things like amyloid beta, which is expressed at the synapses. So this may be how it's happening. So these studies have made a few headlines recently, and there'll be a few things about, oh, how you sleep, the position you sleep to maximize this flow and stuff like that. I've seen an awful lot of stuff online about them. There have been a few comments about this, though. Just noting that a lot of these studies depend on very artificial levels of amyloid beta. We don't actually know what happens at the native level of what's going on here. So it may be we're artificially constructing something which may not reflect what's going on in real life. The other important facet is amyloid beta expression is activity dependent in the neurons, which means during sleep and wake, there's going to be differences in the amount which is actually expressed into the cerebral spinal fluid. So what we may be observing may not actually be to do specifically with the flow. It may also be affected by the fact that we have more of it being expressed and dumped into the cerebral spinal fluid during the day anyway. These are just things which have come out. I don't personally, I believe both can be true, but it has been noted as a criticism. The other thing which these studies lead to is this idea, okay, fine, we sleep more, it's more healthy for us, we'll wash out all these states. It's a nice idea, but there's been an increasing number of meta-analyses of sleep and sleep timings, and they're all showing the same thing, a U-shaped curve. If you sleep between 5 to 8 hours a night, all good. Your levels of dementia, Alzheimer's disease, in fact, death is the lowest it can be if you're looking purely at sleep. If you sleep less than 5 hours, yes, you have an increased risk of dementia, death, all the rest of them, which that then matches this glymphatic clearance. You're not clearing out things that's going to be toxic to the brain. But you sleep 8 hours or more, you're also at increased risk of dementia, death, all these things. Exactly what's going on here? Unknown as of yet. It is unlikely this is glymphatic clearance. This could be a number of potential other factors which is coming in here. But there is something else going on there. And so with that, possibly rather sad message, I will wrap things up because time is creeping on as it does. So, but if I'm going to leave you with anything, I just want to say just take care of your clocks. They're not something we really look after. I mean, I hope I've shown that as we age, we see this loss of function in the clock, where we lose this network. In the eyes, we lose melanopsin signaling. And this can have effects we can have with disruptions to the circadian system. We see changes in brain structure, we see changes in behavior, changes in sleep, where, you know, this is a lifetime value of sleep, and that actually your levels of sleep are important as well. But there's hints where if you restore rhythm, I keep coming back to this grafting the SCN extending lifespan. I'm amazed it's not really been done more. That may be hopefully a grant I'll be looking into. There's hints that actually, if you can restore rhythm, if you can maintain rhythms, if you can maintain these things, it's actually healthy for you. And it has been taken on board. I mean, we are, thankfully, for many reasons, we're well past the 80s, where you have things like Maggie Thatcher running around saying, I survive on 4 hours sleep and I'm fine. And you have, you know, this toxic environment of you need to push through and work through, and it doesn't matter. You can catch up with sleep later on, and you look after yourself. We are past that to some degrees. There's still elements of it. And increasingly, there have been a lot more of these trials, particularly in nursing homes. Not only things like those blue blocker glasses, but things like light therapy, where you give doses of light when you wake up with these light boxes. And these things have been proved to be beneficial, and they can alleviate mood. There are some studies which suggest they can be beneficial for even things like Alzheimer's disease. Some studies, that's still a little bit out there. So, as I say, there is enough evidence that if you look after your clocks, they may look after you. And it never hurts just to be a little bit careful of them. We're not talking drugs, we're talking light, we're talking timing, making sure your meal timings are consistent, things like that. So yeah, I hope I've convinced you somewhat that there are benefits to that, and that somewhat there's a bit of science behind it. So lastly, thank you everyone for your attention. And if you have any questions, I will be right here.
Gareth, thank you very much for a fascinating talk. I'm sure there are thousands of questions.
I hope.
Right, who wants to kick off? Richard.
Well, just the first one, really. When in the old days, when I got jet lag, people said, well, you need to shine a bright light on yourself, in your eyes or whatever, when you get up to resynchronize. They also said you shine a bright light on the back of your knee.
Yes, I'd heard that. How am I going to phrase this? I've yet to see any compelling evidence that bright light on the knees is actually there. It can't hurt to try. Some of it might reach your eyes. I wouldn't say not to do it, but I wouldn't expect any immediate results from it. The light in the face, yes. I'd go, that's a good one to try, but I wouldn't go with the knees.
Listening to the radio the other day, they said that circadian clock is not slightly drift from 24 hours.
Yes.
And that these new receptors, have they recently discovered these ganglion?
Early 2000s.
That sort of synchronizes. That if we were locked in a cave,
Yes.
Clock would
We drift.
divert from the 24 hours.
Yeah. Our human circadian period is a little bit more than 24 hours. A mouse is a little bit less. It depends on whether you're a nocturnal or a diurnal animal. Yeah, so there is a discrepancy. The reason being is adaptability. So much as though that the resonance hypothesis I said that rings true. What's more important, again, at an evolutionary angle, is adaptability. And it is easy. This is now looking at mathematical models and things like that. It is easier to adapt if you are slightly out, because then you've got a little bit of a push to move in that direction, which is also why nocturnal animals are slightly less than 24 hours, diurnal animals are slightly more, because the adaptation is going to be the most common adaptation will be moving it in that direction. We're moving it to later for diurnal, earlier for nocturnal. But yes, it doesn't, if you think about it in terms of the resonance, it doesn't make sense, but in terms of adaptiveness, it does.
Having worked shift work for many years, what
bit.
Not too many hours at a time. Is there an answer to maintaining a healthy work-life balance when you're on permanent night shifts?
It is difficult. Okay, so legal obligation, not a clinician. Anything I say is as advice, nothing more. The blue blocker glasses have been shown to be beneficial for shift work in particular. All combining also that with light therapy, these light boxes. It is incredibly difficult, and no one's hit on anything which works precisely, although Till Ronenberg has, the guy I mentioned earlier, has published a lot and advised a lot on people that can do this. The combination of those, so basically trying to remove light as much as you can when you are not working, and so creating this sort of dark space. It has been shown to be beneficial, at least in terms of mood, in terms of melatonin production. However, yeah, like I say, it's difficult, but things like blackout blinds, things like the blue blocker glasses, things like the light boxes have been shown to be beneficial in a number of studies. Unfortunately, there's no hard fit-all cure, unfortunately, but there are some.
I did hear many years ago whether it was true that NASA were doing experiments with the astronauts. Obviously, there's no light, day or night up there. They tried to turn the circadian rhythm on its head.
I have also heard those, but I'm not aware of them I've not seen anything, but rumors circulated at one point that those things were going ahead. It wouldn't surprise me that they did them, or I just missed them, because it would be the kind of thing which actually would be very interesting to study up there. Yeah.
Were you arguing that we should resist having a dose in the day in order to make
right? Yes and no. Let's go with that. Again, in terms of
occasionally.
In terms of the social jet lag thing, there are studies which said with social jet lag, this idea that you shouldn't actually lie in to recover actually is a little bit beneficial. But in my opinion, those are relatively specific cases. And sleep is so beneficial in terms of cognition, mood, all the rest of the things. There are times when you need to listen to your body, and actually times you just need to sleep. And one of the things which is yet to be studied, which I hope will be studied soon, and the reason I hope that is because I'm putting in the grant proposal to study it, is looking at the effects of these daytime naps, particularly in terms of things like the amyloid clearance that I mentioned at the end. And actually whether things like the daytime naps are not just a symptom of the clock breaking down, but they might be protective. It's not known about that yet, and that's what I hope, if I'll be paid to do it, that's what I hope to study, and that's what we're hoping to get out in conjunction with various other labs at UCL. I would say nap away. I think the effects of lack of sleep are very unpleasant and outweigh any benefits which might be somewhere down the line. So yeah, nap away. It's fine. Often you don't have a choice. I certainly don't.
It's an observation about the human being clock being more than 24 hours. And that is, when I was in my 20s, I and a group of friends went camping in northern Norway in the summer. So we had 24-hour daylight.
Of course.
Camping, we had no other synchronism external factor which was synchronized. And yes, we found our clocks sort of went wrong, and we sort of suddenly discovered it was 2 in the morning, and we felt fine. And the longer we were there, the more it seemed to trip. So, actual evidence I have that what you just said.
So, and also in those situations, you'll be extending the clock even further. But one of the things which came out from these bunker studies initially, what they did was they put people in the bunkers, and they said, right, okay, here you go, here's your bed, and here's your light. People would go to sleep, get up, switch the light on. And because they're switching the light on, the first thing they do is see light. The clock says, oh, right, now's the time to get up. And actually it extends. And you have these weird studies early on where you end up with circadian periods of 26 hours, 28 hours, something daft like that, which was just artificially induced because you're in a process, either in your situation of constant light, or where you artificially create more light just as you're waking up, and that will extend your clock even further. So it's one of those weird confounding things which they didn't work out until a bit too late with some of these initial studies, that's actually what happened. But as you say, there's regions of the world where this happens naturally. And so again, if they just looked at those regions, they might have found this first.
Does caffeine affect the circadian rhythms?
Oh, yes. Yes. So caffeine obviously will wake you up and make you alert, all the rest of it. Russell Foster, I would advise if you're interested in these, Russell Foster's lectures and things like his TED Talks are very interesting. One of the things he often points out is caffeine has a half-life of, I believe it's something like 5 or 6 hours. And so you have to be very careful past midday, because otherwise you are chucking on stimulants which last longer than you expect.
Yeah.
And that it will keep you awake even if you have that cup of coffee at 4, it'll keep you awake longer than you think. And so that will, again, it's another sort of artificial form, I guess, of social jet lag, where you're pushing yourself more awake and more active than you should be. So yeah, I wouldn't advise coffee in the evening, certainly, but yeah, in the morning, just as a kickstart. I mean, I survive on that half the time. But no, it does stimulate. It's more than just sleep. It does also stimulate various circadian cycles as well.
John.
Yes, I was. Do you know if any work's been done for those people who've been in space for many months? Because that must be extremely disruptive. Do they blank out the system?
I don't actually. That's a good question. Again, I presume NASA has these things, and they may be out there. There have been, off the top of my head, I'm aware of papers by NASA. I just can't remember what they did. But actually, it would be very interesting to see how that kicks in, because it also, I mean, there's going to be multiple effects on that as well in terms of meal timings and things like that. So yeah,
because some of them are up there for
many in
months,
the International Space Station. Yeah, they're up there for a very long time. Yeah, something I need to look up.
Yeah.
There were some studies planned, weren't they, for adolescent and school children starting later in the day to try to synchronize.
Again, Russell Foster, there's been a number of pilot studies, and they've shown to be beneficial. That if you start school later, it's more beneficial for the children because, as I showed with the chronotype, they've got a later chronotype. There is then a minor clash, because the teachers have an earlier chronotype. And so there's a minor clash with that. But there were pilot studies certainly went ahead. And I know that came out of Russell Foster's group in Oxford, amongst others. And they got published and they showed results. I don't know where, if at all, that has progressed. It may not have been taken up by anyone because they decided it's unworkable, but it certainly did show some evidence that yes, there is an effect there.
Is there any evidence, Gareth, that tau protein is flushed through with the lymphatic system as well?
Yes. Group in UCL have also looked at tau protein. It's a bit more recent than the A beta, but yes, it seems to be. So same rules seem to apply.
Yes.
We tried experimenting with different hours of work during the day when it's running a business, and we found that you could extend it way down to about 6 o'clock in the morning and way up to 10 o'clock in the evening. And we sort of figured out that you could do that for a while, but then it didn't really add to the performance overall. Would you say that was because we just got shagged and exhausted? Or we're messing with the rhythm?
Both can be true. I suspect, I would suspect there would be an effect on the rhythm, particularly with something like that. You're going to hit sleep problems because there's this idea, okay, I'll stop at 10. Let's go with that, just an example. That's not how your brain will work. Your brain will still take an hour or so to start shutting off, start switching off, particularly if you just come out of, say, work, or looking at a monitor or something like that. It'll take a long time for your brain to actually switch off. And so you will end up leading to disrupted sleep patterns with things like this. There was a very, slightly off topic, but there was an interesting paper by Till Ronenberg where he looked at the Twitter account of a certain Donald Trump. And he identified because Twitter has all the information of the device that was used to make the tweet. And so he could identify when he used his phone, which was his and his personal phone. And he found that he was tweeting at 10, 11 o'clock at night, and then starting at about 6 in the morning. And you can look at that and say, this man is not sleeping. And I know it's a stretch, but you can then look at behavior and start to sort of correlate these things. Obviously, in terms of that, although it was published study, I can't remember, it was one of the Nature publications, but it's, you know, it's obviously a little bit lighthearted, but the same applies, that you are going to be constricting sleep and disrupting sleep. And that's going to probably have a knock-on effect, which will, I think, arguably be a greater impact than actually circadian disruption. Because, as I mentioned, sleep and circadian are separate, and sleep effects are going to be more severe than circadian effects in these regards, particularly long term. You're going to accumulate sleep depth, which will have this cumulative effect on cognition, on things like burnout, stuff like that, stuff like this. So yeah, there'll be a number of factors, but I would guess sleep would be on top.
So is there an optimum length of time before you want to go to sleep when you should stop looking at screens? Is there a correlation?
Depends on your age.
On
your age.
Oh, I'm talking about us.
Again, depends on melatonin, because that will be not melatonin, apologies, melanopsin, because that will be your key thing. It's taking this blue information and shooting it straight to the SCN and these sleep centers and all the rest of it. I don't know off the top of my head for humans exactly what it is. For mice, I can tell you it's about half an hour, but I don't know how that translates.
But you made the distinction when I said that. What about young? Is it different? You
are more sensitive because you have more anecdotally, you are more sensitive, and we certainly know you have more melanopsin expressing cells in the retina. So you are predicted to be more sensitive, certainly. So yeah, I would predict it goes that way around.
One question, Gareth, which I had hoped you might answer, was how is it? It's a subjective thing. Why is it as you get older, time appears to go much faster?
I wish I knew.
Is it because of perhaps our ability to process information perhaps changes again with cell death, and perhaps we're not so quick?
Possibly.
Relatively, information is coming to us faster than we can process, possibly.
Possibly, yes. Albeit, this is now leaning a little bit into more sort of psychological and some cognitive studies. That doesn't necessarily mean you sort of just switch off and you lose time. That that means you might start thinking different things, stuff like that. You might start daydreaming. Effectively. And so your perception time can lose that way. The other thing which does come into play here is again, this is now psychology and other studies, which isn't necessarily circadian. But one thing with our rhythms is we get very used to them. Yeah. And because of that, our brain decides, I don't need to focus on this anymore because I know what's happening. I don't know if, I used to cycle to work and it was the same route every day. And one time, I sort of suddenly almost came to sort of 200 meters down from where I last remembered. It was like, what? Yes. And it was just because I'd done it every day for so long. The brain was just sort of like, okay, I don't need to actually focus. I can just carry on. And that is known to kick in.
I don't think that's true. I think you're focused and you're perfectly alert. You just don't bother the story.
That is more actually.
Deja vu all over again.
No, that's more where it leans into. But whether I'm no expert in that. That's just the small bit of cognitive psychology that I know of some of those. But,
Thank you very much. Oh, one more question.
Quick one. Blood pressure. Taking my blood pressure early in the morning, late at night. It surprised me that after night's sleep, my blood pressure would be high in the morning, low before going to bed. Is that the circadian rhythm anticipating which are going to be active?
I believe so. I'm no expert in blood pressure, sadly, but I believe so.
It seems to be that it's anticipating.
Yeah.
activity.
I mean, so that is,
Anticipating sleep.
yeah, I mean, that is the basis of circadian
Yeah.
rhythms, yeah, effectively. In terms of blood pressures, as I'm no expert, but that would be a reasonable prediction. We have-
Yeah, I think you
Yeah.
pointed out cortisol levels are
Yes,
about
I'd say
45,
core.
aren't they?
Yeah.
Which is surprising because
Yeah.
you think, well,
Yeah.
it shouldn't be
It's anticipating what.
Yeah, because
Yeah.
exactly, you're going to
Cortisol
get
will-
up and start.
Cortisol will elevate the blood pressure.
Yeah.
Depends what sort of day you're going to end.
Yes. Yes.
What's it sleep like that? Or dream like that?
Right. Well, canapes await. So we'll call it a day there. But Gareth, you gave us a very complex and difficult subject, I think, really, and you dealt with it with amazing clarity. So thank you very much. I was particularly impressed with your description of synaptic transmission done in about 3 seconds.
Yes.
The anaesthetist. It was pretty good.
Fair enough.
So
I'm
thank you
across
very
it.
much for an excellent and most entertaining and most illuminating talk. Thank
Very
you.
well.